I don't see Dr. Judd. Oh, there's Dr. Judd. Hey, Nathan, I'm recording, so if you want me to just continue recording, I can post the video when we're done today.
Yeah, that'll be good. I've uploaded the lecture from Wednesday onto YouTube, so it should be available now, if not after class.
Okay, cool.
I did that the day of, but it just uploaded to my channel, not the one that we're doing together. So
Oh,
I had to
gotcha.
move it.
Oh, I didn't even know you had your own channel.
I've been recording some of the ecology classes, and I did some for BD last semester.
Okay, cool.
Okay.
Have to check out Dr. Judd's YouTube channel.
Okay, that's ready. Get my slides ready. Have you done any announcements?
I have not done any announcements. The only announcement I have is that I haven't received feedback from everyone about Apple Pencils and iPads, so I need to know that sooner rather than later so that we can work out a alternative plan over the weekend.
Okay.
So sooner rather than later on that, if you haven't sent that information to me yet. I think that's the only thing on my list.
Sorry. Okay, yeah, I think that I've talked to a few of you in office hours. But today we're going to be doing the central dogma. So we're going to be moving on to another portion of thinking about those 4 premises of evolution by natural selection. And I thought that I would start by giving you all a moment to ask any questions, if you have any, about the homework on population growth from Wednesday. Any things that you feel like cleared up there?
I had a question.
Yeah.
It slipped my mind as to what the lowercase r was, like the definition of it.
Oh yeah. We call that the instantaneous rate of population growth.
Okay.
That's going to differ depending on the size of the population at any given time. Another thing that I think I only mentioned maybe once in passing, I kept talking about the finite rate of increase. That letter is called lambda. So if you want to know what that little Greek letter was, that's lambda, Lambda.
I have a question.
Yeah.
So there was a question and it asked about like the population of tortoises in the future in 2030. Can you remind me what equation we use to find that?
That's the equation where you're going to need to figure out how far into the future and figure out what that value for t is, how many years that is, and see if you can find an equation in that list on the prep homework. I think the prep homework is where it's all listed out, that shows you give a starting population and you want to predict at some value t. It could be any t. It doesn't have to be t plus 1. It could be any t value. Can you see which one that is? The starting population is always with a naught, so a little 0 subscript.
Yeah, I see it.
Okay. Is everyone else good on that one? Had someone else ask a similar question.
Yeah, I'm still kind of confused on how to calculate the little r.
Aha, so that's something that we didn't put on the list of equations, but it's something I was hoping you'd remember from math class. So if you got lambda equals e raised to the r, what can you do to both sides of those equations to get rid of that e? Anybody can chime in here. Riley, I know you know it.
You can take the natural log of each.
Yeah. Take the natural log of lambda and take the natural log of e raised to the r.
And
I
remember,
see-
just for future reference, anytime you do something to one side of the equation, you have to do the same thing to the other side of the equation, just to remind you of that.
Okay, so if that's it for questions, let's see. Now, I meant to give you guys a preparation homework on Wednesday, and I did not do that. So last night I posted, and maybe you had a chance last night or sometime this morning, we had all morning. Hopefully you got a chance to go look at the little video that's posted on the link to the prep homework. If not, just go ahead and do that today. I made the due date through the end of the day today because I didn't give you enough notice on that one. But it's a good way to prepare yourself for what we're going to be talking about in just a minute. And we're going to have a little in-class activity. That's that 18th September Central Dogma activity. We're going to do that as a group or sort of individually, but in class here in just a little bit, depending on how much time we have. So you'll just be able to open that PDF on your iPad or your computer to work it out. Okay. Maximize this. Desktop 2. Okay. Are you all seeing the central dogma slide? Let me make sure that's working.
Yes.
Okay, good. All right, central dogma of molecular biology. Let's refresh ourselves. I want you guys to know these. 4 premises for evolution by natural selection. Make sure you understand what we mean by differential survival and reproduction with respect to some phenotype, whether that's coloration or horn length or beak size or any number of things that might be heritable in a population of organisms. So we're going to be, the next several lectures, we're going to get into this question of heritability and how that works and the genetic basis for phenotypic traits. Today we're going to talk about genes and variation in genes, and that'll lead us to heritability next time. But we're going to focus on this idea of the central dogma of molecular biology. Basically, how do you get from DNA and genes to a phenotype? And we're going to be able to answer a few questions. You'll know what a mutation is in a genetic sense. You'll know about the 3 different classes of mutations and what effects mutations can have on proteins. You'll also be able to think about, you start to connect this to fitnesses of individual organisms. And actually, I don't have the cystic fibrosis example. We're going to do this DNA activity that I posted as the PDF instead. I should change that. First of all, we've got this question. What is a gene? Someone want to help me define what a gene is, or what do you think of when you think of genes?
Traits.
Think of traits, okay. What do you mean? Can you expand on that a little bit, or anyone want to help explain what? So that's a one-word answer. Give me some context for that.
Well, like your genes determine what traits you can have, like physical attributes and stuff.
What do we call those physical attributes? Do you remember another term?
Yeah. Phenotypes, I think.
Oh, nice, excellent. Anybody else? What else do you think of when you think of genes?
Isn't it like from your parents?
Yep. So genes are the unit of inheritance, and you'll hear me say that again later in the lecture. It's what gets passed on from generation to generation. Yeah. Anything else?
It's like the genetic code.
Yeah, it's the genetic code. What, anyone know what that molecule is? What's being encoded, or what is the code? Information is encoded, but how so?
DNA.
Yeah, DNA. So we've got this idea of traits and phenotypes, and we've got DNA, and we know there's heritability, but how do you get from DNA to a phenotype? That's what the central dogma of molecular biology attempts to explain. So here's the definition that I'm going to be using. A gene is a sequence of DNA, some stretch of DNA, that codes for a specific functional product. So the DNA has got to make something. And there are a few options of what that can be. So that's the next question. What are the products that the information in DNA can be used to produce? If you think about expressing a gene, what's the result of that?
Hair color or eye color, different examples of that.
So those are the traits that you get. But I'm asking about, maybe I could have been clearer on this. I want to know about how you get from a DNA code to something like eye color. There's a little bit in between there.
Isn't there a pattern between like TA and GC proteins?
So I think you're getting two things mixed up there, but we're going to talk about each of them. There is a pairing pattern to the units of DNA, right? I said a sequence, though it's a sequence of units, a sequence of nucleotides, and those pair in a particular way. But the products that we usually think of when we think about DNA expressing, when we think about genes being expressed, the product we typically think about is a protein. So DNA, when a gene sequence is expressed, first you have to translate it into RNA, which we'll talk about, and then often that RNA is transcribed into protein. And that protein is what has some activity in your body. It might regulate something, it might have a structural role, it might affect the color of the eye. That protein product is usually what gives you the trait of interest. So what do we mean by this term central dogma? It refers to the flow of information in a cell. You've got DNA as the storage molecule, and you want to get to the trait. There's got to be a flow of information from that storage to the final product, the phenotype. And this is what that means. You'll see that I've highlighted some of these words in a dark red. Those are keywords I want you guys to be sure you know the definition of. So DNA stores information in the nucleus. That information is organized into genes. And those genes are transcribed into a different nucleic acid. So DNA is a nucleic acid. RNA is a different kind of nucleic acid. We'll talk about those differences. And we talk about this word transcribed because you're taking a nucleic acid sequence of letters, A's, T's, G's, and C's, and you're creating another sequence of nucleic acids. It's like a monk transcribing a book in the medieval times, taking the text, making a copy of it. Then, once you have it transcribed into RNA, this little M stands for messenger RNA. It's going to take that information out into the cytoplasm of a cell, and then the nucleotide sequence is transcribed, is then translated into a new language. Instead of the nucleotide language, it becomes the sequence is now in the amino acid language. So you're taking a sequence of nucleotides and translating them into a sequence of amino acids. So going from DNA to RNA to an amino acid sequence that we might call a protein is directional. This is the general flow of information from the cell that gets you from DNA, from information storage to a phenotype. So since you have these three entities, DNA, RNA, and proteins or amino acid sequences, I want you to draw a little concept map linking those, showing the flow of information. Oh, I had 2 slides out of order here. So I'll give you a moment on that concept map. And before I reveal what mine looks like, let's go back to Daniel's comment about pairing. This is a slide that should have come first. This is a cartoon showing how DNA molecules and the genes they're made up of are stored in each cell in our body. So this blue line at the bottom of the screen represents a polymer, a single strand of deoxyribonucleic acid, or DNA. And the letters represent the different forms of those nucleotide units that make up the polymer. Now, a typical DNA molecule is double-stranded. So that means that you've got a backbone here and a backbone here, and it's united where these 2 different letters, these 2 nitrogenous bases, pair up. Notice that C and G always match up, and G and C always match up. And the A's and the T's always match up, whether it's AT or TA. So there's a predictable organization of these nucleotides based on pairing rules. And the pairing rules allow it to coil up into a double-stranded helix. This double-stranded helix is typically wrapped around histone complexes. So these are proteins that are used to wrap the strand up in an organized way. When it's time to express a gene, the 2 strands have to be unzipped so that the different letters can be read by an enzyme. So when it's time to express a gene, those histone complexes relax and open up, and the DNA can unzip, and some other enzymes that are involved in getting that information and expressing it can get in there. These histone complexes, what they do is they allow the coils to then be coiled up again. And ultimately, you get DNA organized into a chromosome. And this is a cartoon you've probably seen before. Now, DNA isn't always in a chromosome. A chromosome is what you get when all the DNA gets packed up, ready for cell division. When it's time to divide cells, you got to organize all the DNA and then allow the process of mitosis or meiosis to occur. Normally, genes are being expressed all the time. Most of the cells in your body have the DNA kind of at least partially unzipped. These histones aren't around all the DNA. It's unzipped so that your genes are being expressed. Proteins are being expressed all the time. But when cell division happens, that's when all this DNA, this chromatin in the nucleus, condenses and gets wrapped up and coiled up into these chromosomes. And that's why you always see DNA represented as X's and lines, these kinds of cartoons.
But those X's, but let me just be clear, it only appears in that X form after it's gone through the process of duplicating the DNA.
Right.
So there's actually 2 copies of the chromosome bound together at the centromere. And so they can appear as just a single line of a chromosome. So the terminology of chromosomes is confusing because you can have a single chromosome.
You'll often hear me call a single chromosome like that a chromatid.
A single chromosome like this, or you can have a single chromosome made up of 2 chromatids bound together here in the middle by a centromere. But they're both called chromosomes, and it's just freaking confusing, unfortunately.
You'll have to grapple with that.
So when we talk to you about mitosis and meiosis, we'll talk to you about the cell phase, and we'll talk about the part of the cell phase where the chromosome duplicates itself.
Yeah.
And so just know that the way we represent them varies somewhat, and don't get too confused by it.
Yeah. So you're looking at a single chromosome composed of 2 chromatids in this cartoon. Okay, so I've got the gallery view up here for you all. So wave your hand or give the little thumbs up or whatever if you drew a concept map for the flow of information that looks like this. See a few? Good, yeah. So DNA to RNA, in this case messenger RNA, to protein. This is the basic graphical way of representing the central dogma. Of course, there's a little bit more to it than this. Just a moment ago, we were talking about cell replication. Whenever you replicate a cell, those chromosomes that condense to form chromatids have to replicate. And then different copies of the chromosomes are passed on to the next generation of cells, the daughter cells. That means that you have to have a mechanism for making DNA from DNA. It's not just DNA makes RNA. DNA has to propagate itself and store that information. So if we label the arrow that goes from DNA to mRNA as transcription, and from mRNA to protein as translation, what could we call an arrow that takes DNA back to DNA? This one is just called DNA replication. So there are certain enzymes that we'll talk about that are involved in making mRNA from DNA. There are also enzymes that are involved in making DNA from DNA. So this is, in some ways, a more complete version. It turns out in the 1970s, we found that there are some viruses that can take mRNA strands, so information stored as mRNA, and insert it into the host as DNA. And so they need a special enzyme to carry out this process called reverse transcription. And we call that enzyme reverse transcriptase. Anytime you add that ASE to the end of the word, you're talking about an enzyme. Reverse transcriptase takes RNA and reverse transcribes it into DNA. And it turns out that some of these RNA viruses can also simply replicate their own RNA. So there's another loop here called RNA replication that's analogous to DNA replication. But no matter what, if you start at DNA, eventually the information ends up over here, either as mRNA, as RNA, or as protein ultimately. Now the reason that I said a gene codes for a functional product is that you'll see in a little bit that there are some products where the RNA is the thing that has a function. It's not always the protein. But usually, and for most examples that we'll talk about, the protein is the functional product. Sometimes the RNA itself is the functional product. Any questions about this concept map or what these different arrows are labeled as? So the next question might be, where does all this stuff happen? Well, transcription happens in the same place where the DNA is because that's what's being transcribed. It happens in the nucleus. And the reason we call this RNA, I often will label it mRNA, is because it's going to take the message coded in the DNA and deliver it to the cytoplasm where the mRNA can interact with another organelle called a ribosome, and that's where you get the translation that occurs. The ribosome interacts with the mRNA to create the amino acid sequence, ultimately the protein, in the cytoplasm. So now you know the flow of information, you know the names of the different steps, and you know where these things happen in the nucleus versus in the cytoplasm of the cell. Now let's talk a little bit more about the structure of DNA. We talked already about how it's a polymer and how there are these base pairings. So let's clear that up a little bit. A polymer is just any molecule that's composed of repeating subunits. In this case, the diagram on the right shows a polymer over here with four nucleotide bases, and then another 4 over here, and they're paired up. Notice the pink and the yellow are paired up, and the blue and the green are paired up. What do you notice is different about the pairing between the adenine and the thymine, the pink and the yellow, versus the guanine and the cytosine, the blue and the green? How do they come together in the middle?
The G and C are like double bonded.
The G and the C, how, can you explain what you mean by double bonded? Because I see 3. So if this one down here, the green one is the G, and this one's the C. 1. Oh, maybe it's just the way it looks on the screen. This is supposed to be kind of like a dotted line instead of a double bond line like this O to the C. This dotted line refers to a hydrogen bond. I can understand if you're looking at a small screen that might look like a double, but in fact these are hydrogen bonds. And hydrogen bonds are going to be really important. You're going to see those again in proteins, and you'll see them when you get to genetics and molecular biology. These are bonds between 2 different molecules. So this nucleotide here, this G, is one molecule, and this C is another molecule. So this is an intermolecular bond, and it's because this hydrogen has a net positive charge as it projects off the nucleotide, and this oxygen has a net negative charge. So it gets kind of sticky and they come together. So it's a bond between 2 different molecules, because some of the elements have polarity. You basically get a negative pole and a positive pole on these atoms, and that brings the 2 molecules together. So the guanine and the cytosine have 3 hydrogen bonds uniting them, and the adenine and the thymine have 2 hydrogen bonds uniting them. That makes sense? You all clear on the hydrogen bonds? Now, if we look at this cartoon version of a nucleotide, I've got base written here. The base just represents those 4 colors. In DNA, the rest of it, this phosphate group and this sugar carbon ring, have a typical structure. And we can label those carbons as 1, 2, 3, 4, and 5. Those are the 5 carbons in the sugar. And then there's this phosphate group here. Notice that the phosphate groups bind to the OH of the adjacent nucleotide. So phosphate, sugar, phosphate, sugar, phosphate, sugar. So these go from the 5' end here to the 3' on the left side, and over here the phosphate is the 5' to the 3' going the other direction. So they're opposite of each other. You'll need to know the 5' and the 3' end in just another moment. Okay, the last term here, the complementary nucleotide base pairing that's held together by hydrogen bonds unites a pyrimidine and a purine. What are the 2 different shapes that you see in those 4 different colors? What's different about this, the base between the pink and the green and the yellow and the blue?
Some of them are hexagons and some of them are pentagons.
So, yes. But let's go a little bit further. So what's different about the ones with pentagons? So here's your little pentagon. It's got a pentagon and hexagon together, right? If you've got 2 rings like that making up the nitrogenous base, that's a purine. And if you've got 1 ring of carbon atoms making up the nitrogenous base, that's a pyrimidine. So pyrimidines and purines always pair up, G and C versus A and T. So we've got pairing rules, and then you've got what kind of nucleotide is it based on the base, the structure of the base. You'll hear me call these nitrogenous bases because these little pentagons and hexagons, they're not all carbon atoms. They have nitrogen atoms in them. So that's why we call them nitrogenous bases. Once you have a polymer like this with the two strands held together by hydrogen bonds, then there's a natural tendency for the molecule to twist and form a double helix. That's why we call DNA a double helix. But if you unwrap the helix, it looks kind of like a ladder, and these pairs make the rungs of the ladder. Another cartoon version showing you a single nucleotide, phosphate group, a sugar, and a nitrogenous base. In this case, it's got the two rings, so we're looking at a purine. Now, RNA has a slightly different structure, but it's still a nucleic acid. It's still composed of nucleotides. What's different about RNA is that it's single-stranded. It doesn't have that double helix. You just get a single read of nucleotides. The sugar is slightly different. It's a ribose sugar instead of a deoxyribose sugar. The other thing that's different is instead of that A, T, C, and G, those 4 possible nucleotides, you have to remember that when you're dealing with RNA, you have to replace the thymine, the T, with a uracil, which we often abbreviate as a U. That's going to be the most important thing to remember about RNA for our activity later. Now, because it's single-stranded, it's got those nitrogenous bases with their little hydrogens, their hydrogen bonding sites sticking out into the environment. So it's pretty typical for an RNA strand to fold and twist into complex structures so that those hydrogen bonds, those polar tips to the atoms, have something to do. That's important because that folding and twisting is part of what makes RNA sometimes the functional product. All right, so now let's go to protein, a little intro to what a protein structure is. If you take 2 amino acids and you put them together to make a polymer, or maybe 3 or 4 or 5, when you put a few amino acids together to make a polymer of amino acids, we call that a peptide. And if you have anywhere from 5 or 10 all the way up to about 50 amino acids in a polymer, we call that a polypeptide, many peptides. Once you get over about 50, roughly, then we tend to call them, we put them in this category of proteins. And there are a bunch of different kinds of proteins. Some proteins have structural functions. Some proteins have transport functions that allow molecules or ions to move across cell membranes. Some of them are, the ones that move across membranes are channel proteins usually. There's transport proteins which can bind to different molecules and help move them around throughout the cell. And then there are enzymes, which are proteins that catalyze reactions. They make a reaction that might otherwise be very difficult or require a lot of energy, require much less energy, and it proceeds more easily. So there's a lot of different possible functions of different kinds of proteins. Proteins are made up of about 20 different kinds of amino acids. In your body, there's about 9 of them that you really need in abundance, but proteins can have more than just those 9. There's up to a total of 20. And that's one of the things that makes the language of amino acids different than the language of nucleotides. In the language of nucleotides, there's only ever 4, A's, T's, G's, and C's. If you get to RNA, then it's A, U, G, and C. And you can put together a polymer in a whole combination of those nucleotides. In the case of amino acids, you put together a polymer out of up to 20 different possible amino acids. There's a lot more choices, a lot more complexity there. And proteins tend to be quite large. So one of the most common proteins on Earth is an enzyme called Rubisco. We'll talk about that when we talk about plants later on. That protein is made up of a polymer of about 477 different amino acids, all folded up to give you this nice product, this enzyme that facilitates a chemical reaction. We'll also talk about voltage-gated sodium channels later and how they work in the nervous system. And a typical voltage-gated sodium channel might have more than 1,500 amino acids making up the whole thing. So quite large, but folded into a compact functional structure. One of the largest proteins is connectin, which is in your muscles. It helps your muscles expand and contract. And that typically has over 30,000 amino acids. in this cartoon, up at the top here where it says VAL, HIS, and THR, those are different abbreviations for different amino acids. So VAL is a valine amino acid, for example. And you can see they're linked together in a chain here. That's the polymer or a peptide chain. And what we find is that as you create a big polymer of amino acids, that polymer will fold and bend into different complex structures. Two really common ones that we call part of the secondary structure of a protein are an alpha helix and a beta pleated sheet. And what I want you to notice about this is that we've got these amino acids that are linked together in a chain here, and then there are hydrogen bonds that bring those two different chains adjacent to each other. So you get these sheets that are chains lined up and bound together by hydrogen bonds, the same kinds of bonds that keep the nucleotides together in DNA. There's lots of structures to proteins. Alpha helices and beta pleated sheets are really common, and they all come together to make a big protein. I'll show you a cartoon of the whole protein in a little bit. So how do you get to those complicated protein structures? Well, let's talk about the process of transcription, that first arrow. When it's time to transcribe a gene, the double helix has to unzip, and then an RNA polymerase enzyme, so a protein, binds to this one of the two strands of the DNA. It binds to the antisense strand. And then it matches up the U's to the A's to the T's, the C's to the G's, and so forth. So what you end up with is an RNA transcript that is identical in sequence to the other sense strand, or the sense strand of the DNA. The only difference is what? What's the main difference between this RNA transcript and the sense strand that the RNA polymerase did not read?
The U's or T's.
Yeah. In the DNA, the U's are still T's, and in the RNA transcript, the RNA polymerase uses those uracils to match to the A's. Good. Now, the reason this is purple, remember that's a ribose sugar in the backbone instead of a deoxyribose sugar in the backbone here. So this thing will read off a gene, and then the RNA transcript is exported to the cytoplasm. Okay, and this is the stuff we just talked about. Usually, you might ask, let me start over. You might ask, where does this RNA polymerase bind to start reading a gene? How does it know where a gene begins? There's two answers to that. One of them is that many genes have a promoter region. So a region of the gene, or a region outside of the gene that has what we can think of as instructions to say, this is where the gene starts, read from here. There's also a set of 3 nucleotides that we call a start codon, and that marks the beginning of the sentence. It's like having a capital letter at the beginning of a sentence that you write. So there's a start codon, and then there's a bunch of nucleotides that comprise the gene. Most eukaryotes have genes with both introns and exons. Can anyone tell me what a eukaryote is?
That's an animal, right? Or like, because prokaryotes are plant cells.
Not quite. So we do have prokaryotes and eukaryotes. That's a good distinction. And animals are eukaryotes. You're correct there. But plants are also eukaryotes. Anyone else? How about Daniel and tell us what a prokaryote is?
Prokaryote lacks the nucleus, doesn't it?
It does. What kinds of organisms don't have a nucleus in their cells?
Sorry?
What kinds of organisms don't have nuclei in their cells?
Like viruses and bacteria.
Bacteria. So viruses, remember, they don't have cells either.
Oh.
So they don't count. But bacteria do have cells and they lack a nucleus. So we call those prokaryotes. But animals, plants, fungi, And a whole wide variety of algae and other protists are all eukaryotes. They have a nucleus. And inside that nucleus, when it's time to replicate, the DNA condenses into chromosomes. So eukaryotes are all the organisms that have nuclei in their cells. And many of them, when you create an mRNA strand, it has regions that are called introns and regions that are called exons. So there's a process that has to happen that we call mRNA processing, where you take out those introns and then splice the exons together. You can think of it as taking a long-winded sentence and you want to convert it into an easy-to-text message. So you get rid of some of those extra words that you don't need and you shorten it, make it clear and to the point. That's removing the excess stuff, the introns, and keeping the important information, the exons. Once that mRNA processing is done and the mRNA is composed just of the exons, then it exits the nucleus where we can move on to translation. So translation, once again, is going from the mRNA to the amino acids. And what's important about this diagram is to notice that while the nucleotides were sort of one-to-one going from DNA to RNA, here when you go from RNA to amino acids, it's a 3-to-1 ratio. So every set of 3 nucleotides is called a codon, and a set of 3 nucleotides corresponds to a single defined amino acid. So UGG always codes for this amino acid called tryptophan, TRP. UUU always codes for, what is that, phenylalanine, I think? GGG corresponds to glycine, UCA corresponds to serine. Now, so far, we've only talked about mRNA, messenger RNA, doing this job of going from the DNA to the protein. Some RNA becomes part of this little organelle called a ribosome. A ribosome is composed of both ribosomal RNA, shown here in peach and yellow, and some proteins, shown here in purple, bluish. Together, this protein RNA complex is called a ribosome, and it has the job of translating from mRNA to protein. So here's how that looks in a cartoon. The mRNA strand enters the ribosome between the large unit and the subunit, and there are these molecules, these folded RNA molecules called tRNAs, that read a set of 3 nucleotides, a codon, and they also match up that set of 3 to a specific amino acid. So as this mRNA gets read through like a ticker tape, the tRNAs are matching the codons to the corresponding amino acid, and this little protein chain, this peptide, emerges, and as it gets larger and larger, it starts to fold and becomes a protein. Here's another view of the same thing. Sorry if it's a little bit pixelated on my screen anyway. But you've got an RNA that's being fed through here, tRNA molecules that are reading those codons in groups of 3 and creating this chain, this growing chain of peptides. So here's a long peptide chain, or I guess we can call this one a protein, and it goes from an amino end to a carboxyl end, and it's made entirely as a polymer of amino acids. Then that's what folds into those alpha helices and beta pleated sheets, and you get this globular structure that we call a protein. And that's what's shown in this model down here. Notice the helices, these arrows represent those beta pleated sheets. It's got a whole lot of complex structure from all that folding. And that's why you think of proteins as little globs, even though they're in fact created as polymers. So you might have noticed that I told you that there were 3 nucleotides per codon and that there are only 20 amino acids. How many possible combinations of 3 nucleotides are there? How many different ways can you put 3 of nucleotides of the 4 possible together into a set. Can anyone see how to do that? If you start out with an A, say if you start out with a first letter A, how many different possible sets of 3, how many different possible codons are there?
$16.
Mm-hmm. And you've also got 3 other starting options. So what does your new total come to if you account for all possible starting nucleotides?
4.
Yeah, 64. Good. That's important. There's 64 possible codons, but only 20 amino acids, which means there's some redundancy. That's why in this cell, for example, you can see that proline is an amino acid, PRO, that can be transcribed from either a CCU, a CCC, CCA, or a CCG. All 4 of these codons have the same end result in an amino acid sequence. They all code for a proline. Now, which position in the set of 1, 2, 3, which position varies among the 4? Position 1, position 2, position 3.
Position 2.
1 is always a C. I think I heard you right. 1 is always a C, 2 is always a C, but position 3 can be any of the 4 possible nucleotides. You'll see that over and over again. Over, let's see, up here in the first section, UUU or UUC both give you this amino acid. What's different is the identity of the third position. Whether there's 2 or 4 or some other number of redundancy in terms of number of codons, it's almost always the third position that varies and gives you that redundancy. So if you get a mutation, a change from one nucleotide to another in the third position is not likely to affect the resulting proteins. But if you were to change let's try this AAU, if you were to mutate the first position to a C, what amino acid would you get instead of AAU? If you mutated the first position in AAU to a C, what would the resulting codon be and what would the resulting amino acid be?
HIS.
Correct. Yep. Good. If you substitute a C in right here, it puts you up here and you get this amino acid instead. Now you'll also notice that there's a green, this AUG. This is the start codon. This is almost always present at the beginning of a gene. It says this is where the gene starts, and every set of 3 after this is a codon. And then when you get to the end of a gene, there's a stop codon. And there's a few different options for the stop codon. It can be a UAA, a UAG, or a UGA. So a few different options for start, or one option for start, several options for stop. Another way to think about why is there 64 possibilities, right? You start at one of these 3, U, G, A, or C, and then whichever one you start with, you've got 4 options for the second position, and then 4 options for the third position, and then some portion of that wedge is going to be the corresponding amino acid. Some of them are only 2 possible codons, some of them are 4 possible codons. And then you can also see the start and stop options. All right, so review. We've got our DNA, we got our transcription, we've got our translation, and that's how we get from DNA to RNA to a amino acid chain, a polypeptide or protein. And then the proteins and their interactions in your body with the cells and other proteins is what ultimately gives you the phenotype, whether that's flower color, eye color, or some gene that influences your height, or any number of other characteristics that we'll talk about. One other term that I want you guys to know about is an allele or an allele. Either way is correct. If a gene is a section of DNA that codes for a specific functional product, often a protein, arranged somewhere in the genome or on a chromosome when it's condensed, an allele are alternative forms of a different gene. So maybe you've got one or two amino acids different, so the protein is slightly different. If that functional product is slightly different but still serves the same purpose to determine eye color, for example, we call those alleles. So here's a chromatid, an unreplicated chromosome, and this is the location on the genome, in this case on a condensed chromosome, for a gene that controls the color of the flower. And this version of the gene, this allele, gives you purple flowers. Now here's a mutation that gives you a slightly different version of that gene, where the resulting protein produces white flowers instead of purple flowers. So you got two alleles, both for the gene flower color. There's a few different kinds of mutations. What I want you to know about mutations is that they're essentially random. Mutations just happen. DNA is being replicated all the time, and errors can accumulate as it's being transcribed, just like in a monk transcribing a manuscript in the Middle Ages. Once in a while, they introduce an error, and you get some confusion about, whether Mary was a virgin or not, for example. So, or something else, right? So mutations are introduced generally randomly, and there's a few different kinds. One of them, the one we've already talked about, is a substitution, where you take one nucleotide or set of nucleotides and substitute in some alternative nucleotides. You can also have what we call an indel, which stands for insertion or deletion. That's where you accidentally add an extra letter in or take an extra letter out. So that would lengthen or shorten the gene a little bit. And then there's duplications. You could have, say you're reading an AGU or some other set of nucleotides, and then you just repeat that again before continuing on. That's a duplicating a region of our stretch of nucleotides. All of those count as mutations, and they all can affect the ultimate functional product, whether that's mRNA, whether that's RNA or a protein. The rate at which these errors, these mutations happen, can vary widely depending on the kind of mutation it is, or the nature of the gene, the type of organism. Organisms with faster generation times often accumulate mutations faster than organisms with slow generation times. So take a moment and look at this little problem here on the left. I've got an mRNA strand starting here with C and ending with A, and I've got a substitution mutation here. And I want to know, if you read this in groups of 3 as codons, is this a silent mutation, or is it going to change the resulting amino acid? What do you
It's
guys?
silent.
Go ahead, Tessa.
It's silent.
It's silent. So what 3 letters comprise the codon that you noticed? The codon of interest.
The CGA to CGC.
Good. Yeah, that's the codon. We skip the first one, go to the second one. That's the one that includes this substitution. And what was the original amino acid?
CGA.
That's the codon. And what's the amino acid it codes for?
I don't know how to pronounce this. Arginine.
Arginine is correct. Yep, arginine. And when you go from an A to a C, it's still an arginine. Good. Can anyone tell me which mutation in this second, more complicated problem, which one is not silent? Which one changes the resulting amino acid?
Is it AAU to GAA?
Yeah, and there's two there, right? Is it the first position or the third position that makes the bigger difference? I guess that's kind of a weird question, but I think you got it.
There's a third. I don't know. I don't really know.
So you're correct in identifying this codon, right?
Yeah.
If you start here at A, U, then you're at asparaginine.
Uh-huh.
You have to start at a G, you're already over here at a valine. Whether that's a U at the end or an A at the end, you're still going to get a valine either way.
Yeah.
So it's that first position that matters. Whereas here you got a third position, it's silent. Here you got a third position, it's silent. And here you got a third position, and it's silent. So often the first and second positions are the ones that make a difference. When you have an insertion or deletion, that can affect which group of three the tRNA molecule in the ribosome reads. So we call that a frame shift. If that reading frame of 333 is shifted by +1 or -1 nucleotide because of an insertion of an extra one or a deletion of one, then every codon is going to be off, and every one of those is going to be potentially a different amino acid than it would have been without the mutation. So a frame shift mutation makes a big difference. And if the resulting frame shift creates a stop codon and the polymerase molecule stops reading, then you're not going to get any transcription or translation. So we call that a nonsense mutation. If it doesn't create a stop codon and instead creates a different sequence of amino acids, we call that a missense. It's still creating amino acids, but not the right ones. Here's what I mean by a frame shift. In the bottom here, you're switching, we're missing this position where it's either an A or a U, depending on DNA or RNA. And as a result, you get a different amino acid than you should have. You get a leucine instead of a I forget what that one is. And you get an alanine instead of a glycine. Whereas here, if you create a stop codon, the amino acid chain is never translated. Okay, so I'm going to switch us over here to introduce the activity, which we'll treat as kind of homework. I want you guys to email me the result of what you get for your activity. So let me check here. We'll come back to this. Yeah, we'll come back to this. Stop here and stop the share. And I want to show you what you're going to be doing. All right, so you should see the Moodle page now. If I'm successfully converting my screen share.
And your desktop.
Say it again?
We're seeing your desktop.
Okay. So this 18th Central Dogma activity, you'll just download that PDF. and you'll see a DNA sequence. Notice it's double-stranded. Here are the two sequences. And what I want you to do is take this antisense codon, or antisense strand, the bottom strand, and transcribe it into the language of mRNA. So you got to know.
We're still seeing your desktop.
Oh, wrong desktop. Sorry, I have two screens up. This is the desktop that I meant for you to be seeing. You see the DNA strand now?
Yeah.
Very good. So double-stranded. The bottom one is the antisense. So this is the one I want you to transcribe. And you're going to write it down here in the language of RNA. Then I want you to find the frame. That means you need to identify the start codon, and it's on that wheel, and it's, which is the second page here. Right? You got to identify the start codon of AUG, and that'll tell you where the frame is. Then draw little vertical lines around each set of three nucleotides that you'll call a codon. And then finally, I want you to take those codons and use the single letter abbreviations for the amino acids. So on the wheel here, if you were to find a codon that says CUU, then you'll write the letter L for the resulting amino acid for leucine. So if you find CUU, you'll write the letter L right here. Does that make sense? The thing to remember here is to find the start codon before you draw your lines around the frames. And you can do this on your iPad. It's a PDF, and I'll put a little spot for you to drop it on Moodle, like an assignment. Normally we do this in class, or you take it home as homework, but I'll put a spot to turn it in on Moodle. Sound good? All right. That's what I have for today. Paul, do you have anything to add?
I do not.
All right, take a stab at this. Pretend you're a little RNA polymerase, and then put your ribosome hat on and read out that amino acid sequence.
When do you want us to have that done by?
I'll put a folder on Moodle that'll expect it in before class on Monday.
Perfect.
All right. Happy studying.